more kind of fate decisions like mesoderm or neural tissue. So there are different stages that we can replicate. There are different kind of complexity of the models, if you like. But all of them are trying to get at this idea of, well, how the cells build embryos and can we learn about that logic with in vitro culture?
And as I understand it, one might think that if you're trying to make one of these later stage embryos, it's going to be more embryo-like because you're getting the different tissues and so on. But as I understand it, and you said that the first thing the very early embryo does is to make those extra embryonic tissues that it needs. So actually, those seem to be crucial to the potential of an embryo model to get very far along this developmental process. Is that right?
Well, I'm not sure we know. I mean, certainly the embryo models that we have at the moment are not embryos, right? At the moment, they often have very stark differences to the actual embryo. If you looked at a real embryo and you looked at an embryo model, you would easily be able to tell what the difference is. They often don't have the right proportions or quite the right form. Certainly functionally, none of them have ever been able to kind of implant, for example, the extra embryonic tissue ones have never been able to implant and lead to anything that's kind of organized, even in a mouse sort of embryo model. So we're still a long way off kind of technically getting something that can progress further. But do you need the extra embryonic tissues in order to get slightly later stages? I'm not sure you do, actually. I think embryo models have shown us that, the embryo does. Certainly for the embryo, it's important to have extra embryonic tissues because it needs to implant before it starts all of those processes. But actually, I think that's been one of the surprises of embryo models, that you can kind of skip certain stages. Of course, embryo models always skip sperm and egg. We skip fertilization because we start with stem cells, which means we kind of jump in the process a bit late. But actually, I think you can even skip some of the extra embryonic tissues because the embryo doesn't rely on the kind of placenta until quite a lot later, actually. It doesn't rely on some of those sort of later processes until quite a lot of the embryos form. So, I mean, I guess it's an open question. And one of the ones that I didn't think we would be kind of answering with embryo models, but I think we actually are. We're showing that the organization of the embryo itself doesn't necessarily have to have the extra embryonic tissues. I think that's been a surprise.
I know we don't know the answer to this question, but I wonder what your feeling was for how far a model like this could get. But in particular, let's say if we have an embryo model of a later stage embryo, so it's developing more of the structure, but it's never going to have the ability to implant at all. Might it be possible to keep that growth growing without implantation? For weeks, months?
I mean, like you say, it's very hard to predict. I mean, I can't see any technical reason why it wouldn't be possible to, rather than having implantation, to be able to provide that support kind of using bioengineering principles, for example, right? We already use incubators to keep our cells in because they're warm and they have the right oxygen and CO2 levels. So long as we're providing these structures with the kind of right nutrients, the right gas exchange, the right pH, all of these kind of things that we have to set up, I don't see any reason why we couldn't add that kind of role as a kind of engineered addition rather than having it be cellular. Yeah. I mean, I guess the bigger question is, can these things self-organize and self-develop for long enough in order to get to those stages? And I don't know that we're there yet in terms of knowing that. I mean, certainly the experiments so far have been going on for maybe a week or so. They don't tend to progress for very long time periods, but I can't say it would be impossible.
Which I guess is going to leave some people, fretting that, oh my God, before long we're going to be growing people in hatcheries like Aldous Huxley. So it's really important to make it clear, first of all, that this is there are regulations for this sort of research, but also that it's recognised that there are ethical considerations that the answers to which are not obvious. So this is being intensively discussed, isn't it? That, how do we think about this work ethically, both in terms of regulation, but perhaps even more broadly and philosophically, how do we think about it? What is the status of one of these entities?
Exactly. And I don't think I have I've been thinking about this for 10 years. I still don't think I have a good kind of verbalized way of thinking about these structures when they get incredibly complex. You know, if it's got human DNA and it's got various cell types that the embryo has, you know, I don't think these things are embryos, obviously, but do they have some sort of special status because they are kind of from human cells? I mean, I don't know. And I think this is a broader question. You know, as scientists, we can say this is the work that we're doing, this is our intention. You know, I think embryo models have got huge potential advances, not just in understanding development, which is, of course, itself a goal that many of us are working towards, but also better understanding diseases, things like congenital abnormalities, better creating drugs for things like sort of in pregnancy. How do you know if a drug that you're using is safe for the embryo? Maybe we could use these embryo models to better understand that, or to improve IVF rates so that IVF becomes much more efficient. So there are definitely clear advantages to these things, and I think that has to be weighed whenever we talk about ethics. We have to weigh, well, what's the application, what's the intention, and what's the benefit potentially that balances out the risk. And yeah, I mean, it does open up a huge number of questions. How do we currently regulate sort of sperm and egg derived embryos, and then where do these models fall into the kind of wider ethical regulatory landscape? Because clearly they're not embryos, but they're not kind of just basic clumps of cells. As you say, they do invoke certain ethical questions. And so how do we address them? Who makes the decisions? How do we have that global conversation about how best to do that? I mean, it's very much ongoing.
Mm-hmm. Yeah, I mean, it's always kind of interesting to me how one of the, when Yamanaka made the discovery about the possibility of reprogramming cells to a stem cell state, one of the points that was talked about in relation to that, first of all, I'm not sure how much for discussion there still is here's a way to do research on human embryology that doesn't require actual embryonic stem cells taken from IVF embryos, which some saw as ethically questionable. That if you're doing this work, you know, with a bit of skin that you've reprogrammed, it doesn't obviously seem ethically problematic. No. But then if that bit of skin is showing the potential to grow into something truly embryo-like, you've got another question you've got to address.
It kind of opens up a lot of this question about potential, doesn't it? I think that's the big thing that I wrestle with. That, you know, an embryo has a potential to become something that has an ethical status. As soon as your skin cell also has that potential, does that change its value? I mean, we shed skin cells all the time. I don't think we actually value the skin cell itself, but once you perform all these processes, you kind of support it in the right environment, then it has a potential again. I mean, it challenges a lot of our assumptions and a lot of our preconceived ideas about potentiality and life and all sorts of things.
And I guess it does so because of the basic biology that it's not as though philosophers are asking new questions and biologists are wrestling with how to think about them. It feels like it's the other way around, that it's because of the discoveries that are being made in the plasticity of the human form, the potential of the cell to become things we never really imagined it could become. That forces some of the new ethical questions and new philosophical questions, like questions about identity and self and individuality. You know, the idea that every bit of me could become a person. Possibly, we don't know if that's true, but it's looking like it's not obviously not true.
Yes.
How do we think about that?
Yeah, it's so hard. And I think, you know, the important thing is that we have the conversation, and the scientists are very much asking for input. You know, scientists like guidance, we like regulation, we like knowing where the boundaries are. And so it's very much that scientists are trying really hard to be responsible. We're trying really hard to say to people, look, we're raising these ethical questions ahead of being anywhere near that kind of point. I mean, I certainly don't think we're anywhere near that now, but can we have a wider discussion about what an embryo is, what value we place on the embryo, and what value we would place on something that's embryo-like, but not quite an embryo? You know, at what point does that blurred boundary line become essentially not there anymore? And essentially the two things are indistinguishable. So it's early days, and I don't want to overstate kind of where we are now. We're certainly not anywhere near anything that I would be ethically concerned about at the moment, but it's worth having the conversation ahead of time.
Well, maybe one philosophical issue that it raises, and particularly if we're thinking about the issue of potentiality of, what this entity could or couldn't become, is the question of goal directedness in biology. And, all of this work that you've talked about really does look like there's a kind of goal directedness, doesn't it, to these entities, that they, you grow an organoid in a dish and it really looks as though it wants to become something.
Yeah.
And that's always been a contentious issue in biology. To talk about goal directedness at all is to, raise spectres of some sort of teleology. But in development, I think this tension has always kind of been there, hasn't it? But in development, it's not only impossible to get away from, you see it happening before you, that, there is a kind of a directionality to the becoming of these things. So, do you feel that it's valid to talk in terms of goal directedness in this sort of research? And if so, where do the goals come from?
Yeah, the second question is the hard one, right? I think how we talk about things, at the end of the day, if the words I use are clear to you and it communicates this idea that I'm trying to get across, then it's useful, right? In some sense, it's useful as far as it frames our thinking and at the level that we're trying to understand something, if it helps us to unpick that, then it's useful, right? So yes, in development, I think we have to, and we often are quite open about the kind of goal directedness that we see as organs form or as tissues form. How do we communicate that? I think about it often as rules, right? That there's nothing, it's certainly not the DNA that is this kind of blueprint, as you said. It's clearly not just that the DNA itself, by itself, is doing this. It's that the cells are somehow using those building blocks and using that machinery in order to start to make decisions. And that, as you say, has to be kind of this multi-level discussion between genes and cells and tissues and organs that goes up and down and influences their decision making. Like I say, I tend to think about rules and logic and networks more than something outside that's being imposed on the cells. But it's incredibly hard to kind of put your finger exactly on how this is happening and what the right terminology is for it. So maybe we turn to people like you for that.
Well, I wonder, in some sense, one could say, well, is it any more goal-directed than, we don't talk about interstellar dust as having the goal of becoming stars, but that's what it does. Right. And that's what the forces that act bring that about reliably. I think what seems to be different in biological systems is the, maybe you could say, the adaptiveness of it, that, during development, sometimes things do look like they are kind of going wrong. A tissue doesn't quite sort of form or pose properly. And sometimes that leads to real problems. Sometimes that means the organism isn't viable. But as I understand it, quite often, the organism is able to compensate, is able, further down the line, is able to do something that takes care of that. So that, for example, if a limb grows shorter than it would normally for some reason, the skin adapts because it feels the tension and there's that responsiveness that is different to what we see in
absolutely. There's this tension, I guess, between what a cell does do and what a cell can do, and I think that's often quite hard. We are often using experiments figuring out what it does do rather than, as you say, perturbing it, changing the environment, seeing what it can do, which is often a huge amount broader than what it ends up doing. Certainly the embryo is what we call regulative. So you can take a very early embryo that's just kind of divided into a few cells and you can literally chop it in half. And both halves will form completely normal embryos. It doesn't care, but it's being chopped in half. And that's not true of every animal species, right? There are certainly some species where if you chop the embryo in half at early stages, you will end up with half of an organism. Mm-hmm. And so this is a really interesting aspect of biology. Why is there some form or organ or decisions that are so plastic and able to kind of adapt, as you say, or regulate, and then some decisions that are much more constrained? And we're just beginning to understand, I think, that logic and those principles, how can we disentangle those two and figure out what's constrained and what's more adaptive?
Right. Well, Naomi, thank you so much. This has certainly helped me to sort of feel like this does feel like a good time to be having these discussions and to be asking these questions and thinking about the right language to communicate in. So it's been fantastically useful.
Thank you. I've enjoyed it too.
Thank you
so much.
Samuel, you may have questions.
Yeah, thank you, Walt. I actually, if we have time, I thought, in light of Dr. Nurse's conversation, I wondered if we could maybe synthesize these philosophical issues. And it seems like a lot of what you're describing, one is related, Phil, to what you describe in the book, which is that life is very different from mechanical systems. And so I think in terms of looking at the broader historical context of biology, and as a philosopher of science, the elimination of teleology was largely thought to be based on empirical grounds. But in developmental biology, Kant himself famously said, you have to treat biology as if there's purpose in organisms. He thought the as if was necessary because he thought scientifically we don't understand how to explain that, but you can't conceptualize an organism. So in biology, there's been a tension, as you were mentioning, Dr. Moris, between developmental biologists know conceptually what you're doing is studying something that's a purposive system. But Kant thought there's a difference between a system that looks like it has purposes that we can't explain how they may be mechanically produced and that really has purposes. And it seems to me a big revolution here in the philosophy of biology that's been created by your and others' work in this field is actually it is the case that these systems, however we describe them, they are purposive. They have a kind of intelligent direction. And this is one of the major differences between life and what we think of as a mechanism. If you smash a computer or cut it in half, there is no possibility that the two parts of the mechanical system can adapt. And that's exactly because there's this option space you're describing in the sort of organisms where they can do so much more. And I wonder if that's maybe a big development in clarifying the concept of life itself is perhaps related to this very big difference between a mechanical function, which there's not a lot of option space around if there's deviation, as opposed to a biological organism where it seems like there's an incredible option space that you can remove some constraints and then see this adaptation. Do you think that's a fair way to think that we've evolved to a point where the mechanical metaphor is really not adequate to think about this, even if we're not quite sure how to cash out what purpose means at this level?
Yeah, that's very helpful. Certainly I think one of the big differences in biology is that the cells are not fixed, right? That they're constantly getting this information and using that to inform their decisions. And as you say, if something happens to its neighboring tissue, maybe that cell makes a different decision. Maybe even once it's made a kind of sort of fate decision, it can change, it can die, it can replicate, it can do all sorts of things. And so it's definitely not static in the sense that some of our kind of machinery is. And as you say, if you break it, it's broken apart. So yeah, I think it's definitely different to the kinds of machines that we would classically think about.
So, and that's why I sort of talk about it in the book as at least at the metaphorical level, probably more than that, thinking about it in cognitive terms is more useful than thinking about it even in computational terms, let alone in machine-like terms. And, we've been doing that all along, talking about decisions and cells can change their minds and so on. And there are some people, including Dan Dennett, who say, well, it's not just metaphorical talk, actually. It's, we're not saying the cells are aware of what they're doing. They're not making conscious decisions, but they are making genuine decisions, integrating information in the way that our, generally speaking, in the way that our brains do. So a cognitive language seems a more useful one.
Yeah. I mean, it's certainly the ones that we kind of default to, as you say. We talk about decisions, we talk about movement and kind of physical directional movement and things. So, like I say, any terminology that helps us think through the problem and helps us to formulate what we mean at the level that we mean it. I mean, obviously that doesn't sort of work for when we bring in gene networks, for example. That becomes incredibly, any kind of metaphor is obviously limited at some point. But yeah, I mean, certainly it's helpful.
I wondered if we could follow up for particularly people listening, maybe about the current regulations, the regulatory framework in science around embryos, because I think that's an important thing for people to understand. What is the current framework? And then just these open questions that you both raised. I think the assumption of current regulations, please correct me, Dr. Morris or Phil, if this is wrong, the assumptions are that there is something generally special about the human embryo. So if we take that as a kind of shared philosophical premise of scientific research, I understand the challenge that's being raised by this exciting work is if you can take a skin cell and basically reprogram it back to such an embryonic state that you can then turn it into an embryonic phase and then into other organs, then, as you said, the difference between the skin cell and the embryo philosophically and scientifically, and then eventually actually, is eliminated. And so then I think we're thinking about, do we have a clear ethical framework in science for how should we think about the distinctive care or value of clearly human life? Knowing now that maybe any tissue that we take from a human, maybe the skin just falls off. Clearly, we're not worried about that. But if you can take it and with all of this material, actually make it like any other part of the human organism and even grow a human theoretically in the distant or maybe not so distant scientific future, is there a way to connect the existing regulatory framework for how we might, as philosophers, citizens, and scientists, think about, okay, should we think there's a special value to human life? And we need to start thinking about that much more flexibly than just embryonic cells or embryonic tissue.
I mean, it's so tricky. I mean, as you know, right, the regulation on actual human embryos is fairly conserved, right? Most countries have adopted what's called the 14-day rule, which is at which embryos can be cultured for 14 days from fertilization, and often used for research within that time period, and then stopped. So that captures kind of the very first sort of stages of development where these kind of extra embryonic tissues are formed, and that kind of internal part of the structure of the embryo becomes the embryo proper, and we start to see this process called gastrulation happening, which is when the first time really that the embryo decides, right, this bit's going to become my head, and this bit's going to become tail, and it starts to form those fate decisions of mesoderm, for example, or neural or endoderm. So that's the current cutoff for actual human embryo research, and that's been in place kind of internationally. Actually, it's been an incredibly successful rule because most countries have kind of stuck with that as the kind of guiding regulatory principle. I think it becomes much more challenging when we talk about embryo models because, of course, we can't talk about it in number of days because in culture, these things can be sped up or slowed down. I've already said we can kind of skip the early time points. We can kind of push the culture to start a bit later. So days doesn't work as a kind of framework. And so then that leads us to say, well, actually, what is it about the embryo that we're valuing? You know, as you say, we can take it as read that we do value the human embryo. But is it the fact that it started from a sperm and an egg that we value, or is it something intrinsic about the structure of it that we value? Is it just that it has human DNA? Is that the bit that we value? Is it a certain feature of it that we value, you know, the formation of the brain or some characteristic that it has? And I'm not sure we've got a really good answer. I mean, certainly embryo models don't fit neatly into the embryo regulation. They kind of need their own new regulation, but exactly what that is and exactly what form that takes, I think is still hotly debated. At the moment, we have a kind of organization called the International Stem Cell Research Organization, the ISSCR, and the International Society for Stem Cell Research, and they come up with this kind of guidelines, if you like, for scientists about where we should draw the lines and which kinds of models should be subject to ethical review or subject to ethical reporting. So that's been incredibly helpful because, of course, science is international. So even if at the UK, if we decided, right, these are our rules, you know, the USA might come up with a different set of rules and France and Germany might come, you know, how do we work together as scientists internationally to decide where these boundaries are? So it's very much sort of ongoing. I think the ISSCR are going to be producing a new set of guidelines any day now. So we'll see. I mean, I think it's a wider question for society as well. What is it that we value about human embryos? And like you say, if we don't think that a skin cell potentially has that same value, even if its potential as shown by PSC reprogramming is that it could go back to an embryo-like state, it becomes incredibly hard to kind of put your finger on exactly why we value embryos and whether that's then applicable to embryo models. So yeah, very much an open question.
Well, thank you. I think that's profoundly helpful to bring to the public, and maybe we'll link to the report when it comes out, the new guidelines. I wonder, just as we're closing, Phil, if you have any thoughts on this. I know you wrote a whole book on how to grow to a human, and you write about this issue in organoids as well in the book.
Well, I just wondered whether it was worth making the point, finally, picking up on what Naomi said there, that in general, scientists are very eager for this to happen, for there to be clarity, regulatory clarity, because, you know, it's easy for sometimes people to imagine, oh, scientists just want to do whatever they like. They don't. You know, you want to know what you can do and what you can't. And obviously, you know, there would be some concern if regulations were extremely conservative and you couldn't really do any of this work at all. But the idea of there being a clear regulatory framework is something that scientists themselves are calling for and that they welcome. I guess it's definitely worth making that.
Yeah, exactly. And I think there has to be that balance, right? I think there's a huge amount of benefit that could be had from embryo models. Because of this 14-day rule, we cannot access human embryos after 14 days. So we just don't know how the actual human embryo starts to form, how it starts to take shape. You know, we can guess from mouse sort of embryology research, and of course that's been incredibly helpful, but a mouse is not a human. And so when we want to understand human disease, if we want to understand human exposure to drugs, you know, we really do need something to plug that gap. You know, some countries have talked about extending the 14-day rule. Should we push that back further and actually allow, you know, sperm and egg derived human embryo research for longer? That's a possibility. You know, all these embryo models might also help us kind of understand that process. So yeah, I mean, yes, we want clarity, we want regulation in place to say, well, there is a limit, but within that, we need to be doing this research, because hopefully that will give us huge insight that could be of benefit to public health.
Thank you. I think that's incredibly helpful, and particularly to show the purpose is we do want to figure this out. The current regulations prevent us from doing it with human embryos, but actually the scientific goal is we want to figure out how the human embryo is developing. And I think even having that clear helps think through the ethical issues. Is we're trying to get to this developmental issue, so we should figure out, okay, if we're going to begin to figure that out, maybe what we're really worried about is something we don't have the current language for. And I think it's very helpful. I thank you very much, and you, Phil, for bringing that out to the public, and maybe even to other scientists and bioethicists. I think we need to stress that when someone at your level, Dr. Moris, is saying we need to think about this, like you're saying, Phil, I think there is a popular perception. People think scientists are these rogues who just want to do anything. And in fact, we hear the best scientists actually saying, please, let's find the right conversation partners and collaborators, because actually we have outstanding questions here that we want guidance on. And thank you both.
Thank you.
Yes.
Thank you.